This is the final installment of Chapter 18. It's part 2, where we continue with the control of eukaryotic gene expression, and then we go into the downstream effects of these changes in gene expression, in particular determination, differentiation, and morphogenesis, which gives us certainly organismal development, but also maintaining tissues like bone and skin and digestive tract, which we cycle through cells during our lifetimes. So all of these processes are connected by this regulation of gene expression. On the surface, these setups can look quite complicated, and there are lots of individual parts. We're not going to break out too much of the individual parts, other than that the key here is whether or not RNA polymerase II can quote unquote land on the DNA and transcribe. And so we have things that will enhance, like for instance an activator will bind in what's called an enhancer sequence and facilitate RNA polymerase II. We also have repressors, and we've talked about repressors in the past. Repressors will also bind enhancer sequences and prevent RNA polymerase II. So we're going to work on some of these pieces, and if you have any questions, please don't hesitate to ask. Because eukaryotic organisms are generally multicellular and can be incredibly complicated in terms of, if you think of a mammalian organism and so forth, we have to have very, very specific and very, very nuanced ways of turning on and turning off genes, or in between, turning them on a little bit, turning them off a little bit. These control elements are known as enhancers. Enhancers are sequences. They're like operators. Remember, the operator was found kind of between the promoter and the gene. In eukaryotic systems, we have enhancers. Notice that there's a break here in the chromosome. That's just showing you that these can be what we call proximal elements, very close to the promoter, or they can be distal elements and literally thousands of base pairs away from the gene itself, even inside the gene or behind the gene as well. We find enhancers all over the place. And enhancers are bound by transcription factors, which are themselves under gene control. So very quickly we can develop a very nuanced way of turning things on and off, and even in between. Maybe a little bit on, maybe a little bit off, dialing it up, dialing it down, creating a more, shall we say, analog system rather than digital all on, all off kind of a setup. So here's a little bit of anatomy of a gene. And again, we're only showing the control elements that are upstream. So we have proximal elements close to the promoter. We have distal control elements, which can be a long ways away. I would point out that we do see control elements even in places like introns and even downstream. So keep that in mind. So proximal means close, distal means far. Here's our promoter. We're going to start transcription here, assuming it's permitted. We will create an mRNA transcript. This is called a primary transcript. It has not been modified yet. It has these introns and exons. Introns are often removed. There are differential splicing paradigms where we have different genes that are spliced differently, so we get multiple types of proteins in one gene. But the long and short of it is that if it's an intron, it can be excised from the transcript. We've got some modification signals here, and once we've done all that modification, you can see now that the transcript is shorter because we've gotten rid of these intron pieces. We've put a cap on the 5' end. We put a poly A tail on the 3' end. The main point of those is to help the mRNA survive. The longer the mRNA needs to survive, the more robust the capping and tail mechanisms are in place. Some mRNAs are not capped and tailed all that much. They only last for a few seconds or minutes in some cases. And so transcription factors are proteins that will either enhance or repress the ability of a gene to be expressed. And since we have tens of thousands of genes, we have tens of thousands of transcription factors. It's one of the biggest families of genes in the human genome are transcription factors. The array of transcription factors is the primary driver for which genes are expressed when. They can initiate, they can help the RNA polymerase land in the right place and express mRNA, or they can inhibit and prevent expression. But they are essential. They really aren't any genes that we know of that don't have some transcription factor playing a role with helping assist or inhibit RNA polymerase. Here's a picture of an enhancer sequence. Again, it can be almost any length. It can even be inside the genes in some cases or downstream. For simplicity, we've just put this one upstream, and you can see that this DNA can be bent back around, and that enhancer sequence can then be brought close to the start site of transcription in a given gene. Many transcription factors not only bind the enhancers but also facilitate this bending of DNA. Estrogen receptor in particular, they when it was first describing the DNA arcing like this, they called it a DNA gender bender. So you have basically two types of transcription factors. You have activating transcription factors. These are ones that facilitate expression. Okay. They are made of two what we call domains. These are functional areas of the protein. One that's going to interact with the DNA, and the second that's going to probably interact with RNA polymerase to facilitate it landing in the right spot on the DNA and transcribing a given gene. Here's a small transcription factor showing you those two domains, if you will. The motifs for binding DNA have been delineated, or many have been delineated. Oftentimes they coordinate with zinc. They call them zinc fingers in some cases. There are other types, but you can see that we have two alpha helices that are interacting with the minor groove of the DNA. This is the major groove right here. These are the minor grooves. And you can see it fits right in there quite nicely. So we have the DNA binding domain that's interacting with the DNA. And then we have the activation domain up here, which would interact with RNA polymerase II or perhaps some other protein that interacts with RNA polymerase II. So here's sort of a quasi animation. We have these activators binding the DNA enhancer sequences. We've got a DNA bending protein that may bring that enhancer around here, and we've got these transcription factors which will bind those activators, all facilitating RNA polymerase II transcribing this gene. And if we have activators, we also have repressors. They inhibit expression of a gene. And again, if the repressors are present, then those genes get shut down. They can act directly on, like for instance, competing for the TATA box on the DNA, or they can act indirectly by influencing the structure of chromatin or so forth, or methylation of DNA. For instance, histone acetylase could be considered that would be an activator if it's acetylating the DNA, or if it's a histone deacetylase, that would then be a repressor. Deacetylating the DNA, the histones, would then cause the DNA to want to clump up into heterochromatin and thus suppress expression. And we don't want to get too crazy here because it's all too easy to start adding things to it. And like for instance, juggling a ball, throwing one ball up in the air is actually pretty easy. Throwing two is a lot more complicated. Then you get 3 or 4 or 5, and the complications go up very, very quickly. To make a long story short, we get this sort of combination effect. We have enhancers, we have repressors, multiple elements, multiple things going on simultaneously to determine whether or not a gene is in fact actually transcribed. So learn the individual pieces, the activator, the repressor, the enhancers, the promoter, etc., etc. So here's an example of two genes, if you will. So we have albumin, which is a very, very important gene made by the liver. It's important for the osmolarity of red blood cells. It's a very important carrier protein for steroid hormones in the blood. And then we have a very specific gene for the lens of the eye called crystalline. And you can see that if you look in the different colors here, that we have a different suite of enhancers. And these are, we're going to just say these are upstream elements. Don't even worry about the internal and downstream. But you can see we have different panels of enhancers. So what will end up happening is that depending on what activators and repressors are there, we might see expression of albumin, but not crystalline. But in a lens cell, we have a different suite of enhancers and repressors, and we get expression of crystalline, but not albumin. And you could see the problem if we start losing the ability to regulate these genes appropriately. That's where we start to get defects. Cancer in particular is extremely an extreme example of where we have deregulation of gene expression. Keep in mind that all cells, whether it's a liver cell or a lens cell, have both genes. A skin cell, which might not express either one of these, has both genes. Whether or not these genes are expressed depends on what transcription factors are out there, what activators, what repressors binding these enhancing elements and or proximal elements around the promoter. Unlike bacterial cells, we do not see operons. Well, there are very few exceptions. There are some out there, but operons are really the domain of bacteria. There are really not very many operons. Like I said, there are arguments for a few minor exceptions in eukaryotic situations. You can have lots of genes controlling a given morphology and phenotype. That's that polygenic and omnigenic aspect of these situations. And again, activators may not just be for one gene. They may activate a suite of genes. If you remember that transcription factor that I showed you where we had the DNA binding domain and the activation domain, that protein is called MyoD. It is a very, very early activator gene, very, very early transcription factor in the differentiation of muscle cells. All muscle cells express MyoD transcription factor. So MyoD will bind whatever gene has a control element site that will affect that gene's transcription. So we have the mechanistic pieces in place now to understand both prokaryotic and eukaryotic differential gene expression. What does this all lead to? I've alluded to it with cancer and some other things. Let's just kind of work through the process. When we have, in the case of mammalian systems, when we have fertilization, the merging of the gametes creates what we call a totipotent stem cell. This totipotent stem cell has a unique set of a unique genome, different from either parent because it's the merging of the two parents. But what it also has is are the transcription factors and regulatory elements from the mother. So we're going to talk about that side of this. So we start with a totipotent stem cell. This is a stem cell that can be create an entire organism. And what we will see is determination and differentiation as we get more and more developed and more specific in the cell types. So what is the difference between determination of a cell and differentiation of a cell? Well, first of all, determination of a cell doesn't manifest as a morphology. It is a commitment step where a cell is going to go down a certain fate. For instance, a totipotent stem cell is completely undeterminated, I guess is the word, except for the fact that there are transcription factors from the mother. We call those maternal effect genes, because those transcription factors are what are going to drive gene expression, which then causes determination. So, but once a cell is determined to a certain fate, then it will follow the process of differentiation. So we see differentiation, which is more of a manifestation of phenotype after determination. It is the result of differential gene expression. If differential gene expression is the process of determination, the manifestation is differentiation. So here are some fertilized eggs. These are totipotent stem cells. And we can see that after a few days, what happens is the cells, there are some determining factors in the cytoplasm. And what happens is these cells will asymmetrically divide. And that asymmetric division leads to different control factors in different sides of the embryo. Like you can see there's a light region and a dark region. As these cells divide, you're going to get uneven ratios of whatever is causing that side of the embryo to be dark and that side of the embryo to be light. And that leads to differential gene expression in embryonic development. So the cytoplasmic determinants, these maternal effect genes, when we have fertilization, I want you to notice the symbols here. We have little circles and little triangles. Notice that they're not evenly distributed across the cytoplasm. They have been localized using cytoskeleton, motor proteins, you name it. What we have is an asymmetric egg cell. That's true whether we're talking about relatively primitive animals or very complex animals. We get an asymmetry of what is inside the cytosol. And so we get fertilization, and the first thing that happens is we get a cell division. We certainly duplicate the genome, but if you look at the cytosol of the resulting cells, the daughter cells, you can see this one has a preponderance of circles. This one has a preponderance of triangles. These two cells are going to have different fates downstream. Those circles and those triangles are transcription factors and regulatory elements, determinants, and eventually will lead to what we call inductive signals. I'll talk about inductive signals here in just a second. Inductive signals are, shall we say, diffusible signals, sometimes cell surface, molecules. There are those out there that are bound to the plasma membrane, but here we're showing a diffusible signal. An inductive signal is where you have one cell during development that releases a molecule that affects gene expression in the other cell. This is basic cell signaling. We have a signal. We've got a receptor. The receptor leads to transcriptional differentiation and leads to gene expression. Slow genetic response. And as we proceed through induction, we get differentiation, and eventually we get morphogenesis. So determination sets the cell on a certain expression pathway. Differentiation is the manifestation of that process. So when we differentiate, we go from a stem cell to a well-defined muscle cell or a fat cell, et cetera, et cetera. Differentiation. And then morphogenesis is when we have induction events and we begin to get three-dimensional structure in terms of tissues and organ systems. So this E stands for embryonic day 8 1/4, 8 3/4. You can see there's a dramatic change in the shape and distribution of these three different cell populations. That's the process of differentiation and induction. This red tissue right here may be sending signals to tell the other parts to do something different. and vice versa. There's a lot of crosstalk here. By the time we get to about a day later, you can see that there has been substantial morphogenesis in this particular organ system, which may involve, looks like heart and gut, liver, the entire internal organ structure of a given organism. The main thing to remember is that induction is talk between cells. One cell, an inducer cell, is releasing a molecule, whether that's a diffusible molecule or it's present on the cell surface, a contact-dependent signal. If it's on the cell surface, that's what we call contact-dependent, because these cells have to contact each other. In this case, though, it's a diffusible signal, and back to our signal transduction. If that cell has a receptor, it will then undergo some kind of a response. It can be a fast cytosolic response, or it can be a differential genetic response. In either case, it's causing the responding target cell to change its behavior and genetic expression. We've got an animation here, and I would strongly recommend you download the PowerPoint so that you can watch and hear the audio with this animation. Remember that we can have fast cytosolic responses, but mostly when we're dealing with embryonic and morphogenesis is we're dealing with transcription factors and mRNA, differential mRNA expression leading to differential protein expression. That's the predominating response when we're talking about embryonic development and morphogenesis, but very early when we have super rapid division, we often have just rapid cytosolic situations. And link the mechanisms we talked about earlier in the lecture to that genetic response. We get some sort of activator transcription factor that's bound with a ligand that then will bind the enhancer elements, recruit transcription factors that then facilitate RNA polymerase II to transcribe a given gene, or vice versa, repressors. And here's a nice little GIF showing the aspect of induction, where this tissue may be sending signals that cause differential gene expression, determination, and differentiation in other cell populations. So it's oftentimes it's diffusible, occasionally contact-dependent signaling. This video is absolutely stunning. It's an actual time lapse of the development of an embryo. And the movie is called Becoming. It won several awards because it's absolutely stunning. So I would really recommend you take a look at this video. I've posted it in Brightspace for you. It's down here, this Becoming. It's worth watching because it will help you understand and appreciate why we are so interested in the mechanisms of differential gene expression. Everything from helping prevent birth defects like my high school friend, or severe birth defects like the Elephant Man, quote unquote, the story of John Merrick, or preventing and treating cancer or other types of genetic disorders. Understanding differential gene expression and how that gene expression plays out in determination, differentiation, and morphogenesis is a major aspect of what cell biology is all about. So thank you for your time. Again, if you have any questions, please do reach out to me. Thank you. This concludes part 2. We will see you in class.